All right, guys. I'm here to do something that's uncharacteristic of me that you don't know that I do. Um, One of the things I do is study life and biology, and it's a more recent interest of mine. So I've been always interested in figuring out how we can manipulate biology to do things like live forever. And other things like that, very esoteric uses of biotechnology to create superpowers or anything that you might think of or have stray thoughts about, and just knew that it really was in the realm of science fantasy, or maybe it wasn't. So anyway, what I've decided to do, because originally I wanted to create a while ago a series of videos on this subject when I became an expert on it. But the thing is, there's so much information that I think that I don't need to demand perfection of my interest. I can do this little by little over time, and so that's what I'm going to do. I'm going to start a series, and I was also inspired by the many great YouTube biology videos that I've been seeing, and how much it's been easy for me to learn about biology in depth, specifically microbiology and cellular microbiology and genetic manipulation and things of that sort day by day. Just to be clear, I really started at the basics, and in fact, I'm not ashamed to say I started with Khan Academy, and I started with Udemy, and I started a few years ago, actually, kind of here and there, looking into and learning microbiology with the intent of becoming an expert microbiologist. In my spare time, I guess. I don't have time to do it with my full-time career at hand currently, but I figured what I can do is I can bone up little by little every day to become an expert. But it was only kind of a pipe dream at that point, until I realized that this is probably something that's in line with one of my greater purposes in life, so that I can discover what the secret of life is, and all that cool stuff. But specifically, how to stop cellular senescence and cells dying. That's something I really want to get into later, like understanding senescence and death. It's truly a fascination of mine, and I think the only way I'm really going to do it is understand the mechanics, the cellular mechanics behind it. I mean, I was one of those kids who was really, a long time ago, into sorcery and all that stuff, and all into the metaphysics and the other really crazy kinds of things that were associated with death and senescence. But I realized that that's only going to get me so far, and being able to one day find a spell or something, or some sort of mystical item that allows for this eternal life type of stuff to happen, or even superpowers or anything like that on the biological scale, is just wishful thinking, because even if something like that does exist, it's pretty far out of the common man's reach. The ability to manipulate technology is, however, the closest thing to the common person's reach. So with that in mind, I'm creating this series of videos, and which I'm intending at this point to document my discovery. So I'm hoping that that will help other people too, and help me. Okay, great. So let's start with what I'm learning today. So today I learned about dyneins, okay? And anyone watching this, by the way, who's an expert microbiologist and is likely scoffing at me because of my current skill. Remember, everyone started somewhere. So I may be starting at close to zero, but I have what I consider to be a tenacious spirit in this regard when it comes to research and learning things. And I will learn what I need to learn to get where I need to go, and we will catch up eventually. And thank goodness for Google, right? I mean, yeah, and other search mechanisms that provide access to information that's less likely found on Google. So I'll catch up to you if you're scoffing at me, and we'll see who has the last laugh. Anyway, So I learned about dyneins today. And what are dyneins? Dyneins are motor proteins. Motor proteins are a class of molecular motors that can move along the cytoplasm of animal cells. They convert chemical energy into mechanical work by the hydrolysis of ATP. So if you've ever seen motor proteins in action, those of you who are not expert microbiologists, there's some crazy videos online about how they work, and people have gone to a lot of effort to put in these really interesting 3D visualizations. I don't want to play a whole video on that, but maybe I can show you what it looks like. Yeah, so haters gonna hate. This is a motor protein. This is literally what happens in a cell. Okay, it's really crazy, but this motor protein is actually transporting. I think this is a microtubule, and this motor protein is a dynein, which is actually transporting a large viral cell or something. Okay, this is a kinesin protein, a kinesin, which is moving. Okay, that may be a kinesin, but it's moving a large cell. And the thing is, these parts, these legs, are actually called motor domains. Okay, so the dynein actually has several parts to it. And I'm learning all these parts, okay? I'm going to jump around, but I mean, yeah, that doesn't look right, but whatever. Okay, these legs are what walk around microtubules. And if you don't know what a microtubule is basically a structure within a cell which is responsible for transport of material within the cell from, for example, the inner part of a cell, the negative domain, to the outer part of a cell, the positive domain. So these are like microtubules. This is the nucleus, and this is the cell membrane. Okay, so all sorts of things get transported along this microtubule, but this transport action is not a passive transport, which means it doesn't just happen on its own, like some kinds of diffusion. This kind of active transport occurs by the use of these dyneins, right? And I love paint. Dynein. And I learned today that dyneins are actually a very humongous class of molecule. Like, these are massive molecules. You'd think, I mean, these microtubules are obviously massive, but I mean, the structure is pretty regular of the protein called tubulin, which comprises these microtubules. But what is interesting is that the dynein itself, which sits atop the microtubule. And by the way, kinesin and dynein. Generally speaking, there are several kinds of motor proteins throughout a cell. I mean, let's see if there's other ones. There's other interesting ones. Look at these. These are myosin, I believe, and these are walking across another type of filament structure called a microfilament in the cell, which is comprised of actin. So in your muscles, you have tons of these things moving along and working constantly. So isn't that crazy, right? I mean, this is another interesting GIF of a motor city protein. Okay, another motor protein. The interesting thing is the complexity behind these motor proteins is insane, right? These are at the nanometer scale, and these things that comprise them are just atoms, right? And like, thousands and thousands of atoms comprise them, but they can actually assemble to form this thing which uses electromagnetics, right? That's how this thing is moving, right? It's electromagnetically binding, and it's using chemical hydrolysis to let go of ATP and move across this thing, right? It's pretty insane, right? Kinesin. This is the domain. This is how kinesin looks, actually, right? It's kind of crazy. I mean, all of these alpha helices, if you're familiar with protein structure, alpha helices are actually one type of polypeptide structure that comprises a protein. Okay, and these little helix corkscrew things are alpha helix structures, which are basically linear sequences of amino acids. I'm not going to go into amino acids right now, and I might go into another video on amino acids later on, but it's insane what a little bit of complexity can do, right? this is replication of DNA polymerase, right? But that's a whole different thing. I just think for now focusing on the motor protein by itself is interesting. This is example of two motor proteins that cause motion of one tube structure across another tube structure. Okay, that's pretty insane, because this is essentially how muscle cells work, right? And other cells that require sliding of these tubes, they have motor proteins attached to each other, which push one tube again relative to another through their motor action, and that's how motion works, right? And I'm sure that many of these motor proteins get dislodged or break over the course of a single sliding action. Think how many thousands and millions of sliding actions every second you have in your entire body of these motor proteins moving microtubules from one place to another, from the positive end to the negative end. It's kind of crazy. Another example of a motor protein, more general, like I guess cartoony example. Anyway, so where was I? Yeah, so let's get back to the microtubules. So the microtubules transport all sorts of things. They even transport viruses, right? Because viruses try to infect the nuclear membrane. Like, for example, the coronavirus it's not a retrovirus, I'm sorry. I'm trying to sound smarter than I am right now, but it's an RNA virus, which attaches via its little spike proteins to a cell, and then through a process called endocytosis, and in some cases, I guess, there's a pinocytosis, which is called another way of saying it is cellular drinking. The contents of the RNA coronavirus, for example, genetic contents will be absorbed by the cell, and it'll be taken down into the cell itself, where the other parts of this virus will replicate through replicating RNA and be sent out of the cell. So, and essentially it will use that to create more genetic copies of itself, right? More coronavirus. So I'm sure what happens is that the immune system and the natural innate responses of cells, I guess, kill some of these viruses. Though if they're not like T cells or anything like that, or B cells, then they won't be able to, I don't think they'll be able to kill these viruses outright before dying themselves. But the few that they can catch are probably transported out of the cell through these microtubules. So I don't know all the details. I'm still learning, but that's kind of essentially what happens. Whoa, what's that? So let's go back to motor proteins. Motor proteins. So, what else is interesting about motor proteins? Best prominent example of a motor protein in the muscle cell is myosin. And so the best prominent example is myosin. You're going to hear the actin-myosin sliding theory, I guess. I forgot what it's called exactly. But contraction of muscle fibers in animals. Motor proteins are driving force between most active transported proteins and vesicles in the cytoplasm. So this is what I was talking about, active transport. That means it's not passive. It doesn't happen in its own. Stuff is actively transported out of the nucleus and into the nucleus via these motor proteins that are acting to transporting through microtubules. And these are all in the cytoplasm of the cell, which is basically the plasm where everything kind of, in the cell lives, like organelles and things like that. And all the types, we didn't even go over the types of structures in cells, but I'll go back to my handy paint program. Discard. So I love this. This paint reminds me of a paintbrush from when I was a kid. It's a great program. Anyway, so there's all different kinds of things in cells. For example, in a cell, you have on the outer part of the cell near the plasma membrane, where you have these microfilaments, which are responsible for a lot of different things in the cell, but they're like the smallest, lowest order structural structure in a cell, which nucleate along the plasma membrane of the cell. When I mean nucleate, I mean they start growing in a crystal form, right? Like, because that's how a lot of proteins grow, and structural proteins, as crystals do, that they additively combine like crystals do. And they start from a nucleus, right? Their own nucleation point, not the nucleus of the cell. Then you have these are the smallest 7 nanometer, microfilaments. Then you have the largest structure, which is called microtubules. That's what we were just talking about. And those, like I said, they go throughout the cell, they transport stuff from the nucleus, and they're responsible for a lot of things within flagella and cilia. So, for example, you have a flagella, which is a motor structure for certain cells, like a sperm cell or something like that, that'll help it move. And those flagellas have microtubules in them, like this. Okay. Not a very accurately drawn cell, but that's what I got. And then these microtubules do all sorts of things, right? Like I said, through the course, the use of dynein. And then there's also intermediate filaments, which are interesting because intermediate filaments they're in between the size of microtubules and microfilaments. And microtubules, by the way, are strangely named. They should not be named microtubules. I mean, maybe they should be, but they're the largest of these structural infrastructure cytoskeletal proteins, right? So I don't know, micro is kind of strange, but I guess it compared to a large tube or something. But intermediate filaments are in between the size and diameter, and they kind of allow for the cell to stretch because they're in an element, a cytoskeletal element, which resists tension on the cell. So basically, if your skin is stretching, the reason your skin can stretch when you're doing exercise and not just tear is because you have intermediate filaments there, which prevent the tearing. Intermediate filaments. Okay, and then you have microfilaments, you have intermediate filaments, you have microtubules. Microtubules you can remember that because intermediate filaments kind of crisscross within a cell. The microtubules kind of extend to the outside from the center, and it's like this starish kind of pattern. And then you have the microfilaments, which are near the nucleus, but they're in many other places in the cell. Also, yeah. So that's, I think, all that I'm going to do today. But we'll get more into different topics as I learn about them, and as they get closer to understanding biology. Okay, see ya.